Are you ready to dive into some serious plant science?
I am. Let's do it.
Today, we're going deep on photosynthesis.
One of those processes that we all know about.
Yeah, how many of us actually understand what's going on?
Yeah, it can get complicated.
So to help us out today, we've got excerpts from a biology textbook,
Great resource.
and we also have all of your notes. You're prepping for a big presentation on plant biology,
Awesome.
and we're going to help you really own in that room.
Exactly. Get ready for some aha moments.
Yeah. Even if you think you know photosynthesis, we've got some stuff here that will surprise you. Even seasoned botanists.
For sure.
All right, let's kick things off. So you already know the basics. Plants use sunlight to make their own food.
Right.
That's what makes them autotrophs, those incredible self-feeders.
The foundation of the food chain.
Yeah, the real MVPs of the natural world, right? But here's something you might not have thought about. Photosynthesis isn't just about leaves.
Not at all. Think about a cactus. The leaves are spines.
Mm-hmm.
So the stem has to do the photosynthesis.
That makes total sense.
Any green part of a plant can do it.
That's so cool. It really shows how adaptable they are. But the thing that blows my mind is how essential photosynthesis is for all life, not just plants.
Right. Without plants, there wouldn't be any food for us heterotrophs.
No burgers.
No salads. No snacks.
And even more importantly, we wouldn't have any oxygen to breathe.
Absolutely critical.
Talk about a party foul. No oxygen, no more podcasts, no more music, no more anything. Really puts into perspective how much we rely on plants for, like literally everything.
It does.
And you know, our understanding of photosynthesis wasn't always so clear. There were some fascinating early experiments that paved the way.
Absolutely.
Have you ever heard of Joseph Priestley?
Of course. The guy who discovered oxygen.
Right.
Yeah, in the 1770s, he did this really neat experiment. Put a candle and a mouse in a sealed jar. And of course, the candle went out and the mouse, well, didn't make it.
Yeah.
But then he added a mint plant to the jar. And guess what? The mouse survived.
Whoa.
And he could even relight the candle.
Wait, how do you relight the candle without breaking the seal? Did they have like extra long matches back then?
I don't know about the matches.
Yeah.
But what's important is that he showed that plants somehow restored the air.
Oh.
It paved the way for understanding the link between plants and animals.
That's pretty amazing. It's like this seemingly simple experiment with a mint plant changed how we view life itself.
For sure.
So what came next?
We got Ingenhousz built on that in the 1790s. He showed that this restoring power of plants only worked in sunlight. He also noticed bubbles forming on the green parts of plants in sunlight,
Bubbles.
which we now know as oxygen.
Wow. Okay, so light is key. But how did they figure out what else was involved?
It was a series of breakthroughs. So you have Julius von Sachs, who proved in 1854 that plants produce glucose,
Okay.
stored as starch
Right.
during growth. And he figured out where it was happening. Those chloroplasts.
Ah, those little green machines.
Exactly.
Okay, so we've got plants using light to make glucose and oxygen, and it's all going down in the chloroplasts. But what about the other pieces of the puzzle?
Right.
Like, where does that oxygen actually come from?
That was a big debate for a while.
I bet.
The early equations for photosynthesis were all about carbon dioxide. But then Cornelius van Meel, in the 1930s, figured out the released oxygen actually comes from water,
Water.
not CO2.
That's a game changer. Okay, so 6 molecules of carbon dioxide plus 12 molecules of water in the presence of light gives you 1 molecule of glucose, 6 molecules of water, and 6 molecules of oxygen. That's a mouthful. But you've mentioned before that it's not just one reaction, right?
Oh no, it's a whole chain of events.
Okay, so to understand it, we need to zoom in on those chloroplasts where the real magic happens.
Exactly.
All right, let's grab our microscopic lenses and get ready for the close-up.
Let's go.
Take us inside those chloroplasts.
Okay, so picture this. Inside a plant cell, you've got these tiny green organelles, the chloroplasts, and they're packed with this complex membrane system. Grana, stromal lamellae, and the stroma.
Lots of as.
Yeah.
And they all have different roles.
Each one plays a specific part in this whole photosynthetic dance.
It's like a well-choreographed ballet happening inside these tiny chloroplasts.
You could say that.
But hold on, how do these chloroplasts actually capture that light energy? I know chlorophyll is involved, but is it really the only pigment doing the work here?
Why do you think there are so many shades of green in plants? Why do leaves turn yellow and orange in the fall?
Okay, so you're saying there's more to the story?
There is.
Those other pigments are involved in photosynthesis too.
You bet. So besides chlorophyll A, the main player,
The star.
yeah, you've got chlorophyll B,
Okay.
and then a whole supporting cast of carotenoids and xanthophylls.
Whoa!
Each one absorbs light at slightly different wavelengths.
Ah, so it's like widening the range of light energy the plant can actually use. It's like having an all-star team of light absorbers, each specializing in different wavelengths. This is way more complex than I imagined. But how does a plant actually use this absorbed light energy to do something useful?
That is a great question, and that's where the electron transport chain comes in. Think of it like a cellular relay race.
A relay race. Okay, I'm intrigued. Tell me more. What exactly is being passed in this race?
It all starts with photosystem 2, PS2. It absorbs light,
Okay.
energizing its electrons, and then these energized electrons are passed along a chain of molecules, like runners handing off a baton.
I love that analogy.
Yeah.
But wouldn't this electron relay eventually run out of steam? How does the system keep going?
That's where it gets really interesting.
Here we go.
The electrons that PS2 loses,
Yeah.
they're replenished by splitting water molecules.
What?
Yeah. And this is crucial because it provides the electrons to keep the relay going,
Okay.
but it also produces the oxygen we breathe.
Wait, so the oxygen we breathe is basically a byproduct of plants splitting water.
It is. Pretty neat, huh?
That's mind-blowing.
It's one of nature's most amazing tricks. But there's another crucial outcome of this water splitting. It creates a proton gradient,
Okay.
and that drives the production of ATP.
Hold on. Proton gradient, ATP. There's some pretty technical terms.
Yeah.
Our listeners might need a little help understanding what they mean and why they're important. Can we break it down?
Sure. So those protons that are released when water split,
Yeah.
they don't just float around. They accumulate inside the thylakoid lemon,
Right.
creating a higher concentration there than in the stroma.
So it's like blowing up a balloon, creating a pressure difference. But how does this proton pressure translate into ATP, the energy currency of life?
Okay, well, that's where this remarkable enzyme called ATP synthase comes in.
Okay.
It's like a tiny turbine embedded in the thylakoid membrane.
Wow.
And as protons flow through ATP synthase down their concentration gradient, the enzyme spins.
Okay.
It captures that energy and uses it to create ATP.
A tiny spinning enzyme creating the energy that powers life.
Isn't that cool?
That's incredible. It's amazing how much complexity is packed into these tiny chloroplasts.
That is.
And all of this, the electron transport chain, the water splitting, the ATP production, those are all part of the light-dependent reactions.
Correct.
But there's another set of reactions waiting in the wings, the Calvin cycle.
Exactly.
Okay, so this is where the carbon dioxide gets fixed and those sugars are built, right?
Yes.
But didn't you call those the dark reactions? Does that mean they only happen at night?
No, not at all. It just means they don't directly need light.
Ah, okay.
They use the ATP and NADPH, another energy carrier, that were produced in the light-dependent reaction.
So it's more about energy flow than actual darkness. Light reactions capture and convert the energy, and the Calvin cycle uses that energy to build the sugars that fuel life. It's a perfect partnership.
It is.
But I'm guessing there's a lot more to this sugar building process.
Oh, there is.
Let's dive into that Calvin cycle and see how those sugars are actually made.
All right, let's go.
Lead the way. I'm ready to get my hands dirty in the sugar factory.
Let's do it. So imagine a bustling factory with assembly lines, specialized workers, all focused on building sugars from carbon dioxide. That's the Calvin cycle.
Sounds busy in there. So where does the carbon dioxide enter this sugar factory?
It all starts with a 5-carbon sugar called RuBP, and then an enzyme called Rubisco grabs a CO2 molecule from the air, attaches it to RuBP.
Okay.
That creates a 6-carbon molecule that quickly splits in two. You get 2 3-carbon molecules called PGA.
So Rubisco, the master builder. But I've heard that Rubisco has a bit of a split personality. It's not always the most reliable worker in the factory, is it?
Yes, that's right. Rubisco, it can sometimes grab an oxygen molecule instead of carbon dioxide.
Uh-oh.
And that's when things go a little haywire, leading to a process called photorespiration.
Photorespiration? That sounds counterproductive. Isn't the whole point of photosynthesis to use carbon dioxide and not release it?
You're right. Photorespiration is like a detour in the sugar factory. Consumes energy, releases CO2 instead of fixing it.
Not ideal.
Not ideal. But scientists think it might have some protective functions for the plant.
So it's like a safety valve, maybe preventing damage under certain conditions. But I've heard that some plants have evolved ways to actually minimize this photorespiration.
Exactly. That's where the C4 plants come in.
Ah, okay.
They've got this clever strategy to concentrate carbon dioxide, making Rubisco's job much easier.
The C4 plants, the efficiency experts, tell me more about how they manage this carbon dioxide concentration feat.
Okay, well, they have a special leaf structure. It's called Cran's anatomy. And in this structure, the bundle sheath cells, those cells surrounding the leaf veins,
Right.
they are enlarged and packed with chloroplasts.
Ah, so it's like a dedicated carbon dioxide pumping station?
You could say that.
So it's like a division of labor within the leaf. The mesophyll cells capture the CO2, and then they shuttle it over to those bundle sheath cells for processing.
Exactly. So in the mesophyll cells, an enzyme called PP carboxylase grabs the CO2, attaches it to a 3-carbon molecule, and that creates a 4-carbon compound. And then these 4-carbon compounds are transported into the bundle sheath cells where they release the CO2. And that creates that high concentration that keeps Rubisco happy and working efficiently.
It's like a CO2 delivery service, ensuring that Rubisco has a steady supply of its favorite ingredient. No wonder C4 plants can thrive in hot, sunny environments.
They've got the system down.
They've got it figured out.
Yeah. But even with all these amazing adaptations, there are still things that can influence the rate of photosynthesis.
Right.
It's this balancing act between the plant's internal capabilities and what's going on in the environment.
It's like a recipe with multiple ingredients, each one playing a crucial role. But what happens when one ingredient is in short supply? Does the whole process grind to a halt?
That's where Blackman's law of limiting factors comes in.
Okay.
It basically states that the rate of a process is limited by the factor that's in shortest supply. Think of it like a barrel with staves of different lengths. The amount of water that barrel can hold is limited by the shortest stave.
That's a great visual.
Yeah.
So even if a plant has plenty of sunlight and CO2, if the temperature is too low or water is scarce, photosynthesis is going to be limited.
Right.
It's a reminder that everything is connected, and even the most efficient system can be held back by external factors. So let's break down some of those external factors, starting with the most obvious one, light.
Okay. Well, light is the energy source, and both the quantity and the quality of light matter.
Okay.
Light intensity, that's the amount of light energy hitting the leaf,
Right.
directly affects the rate of photosynthesis.
Right.
But there's a limit to how much a plant can use.
Of course.
At a certain point, more light isn't going to lead to more photosynthesis.
So it's like trying to fill a glass that's already full.
Exactly.
Any extra just spills over.
Yeah,
But what about the quality of light? Does the color of light matter?
it does. Remember we were talking about the different pigments absorbing different wavelengths?
Yes.
Well, red and blue light, those are the most effective wavelengths for driving photosynthesis.
Mm-hmm.
Think back to Engelmann's experiment. Those oxygen-loving bacteria clustered around the algae exposed to the red and blue light.
So it's like having a radio tuned to just the right station to get the best signal. But even with the perfect light, plants still need carbon dioxide, the essential building block for those sugars. How do they actually capture this vital ingredient from the air?
They have these tiny pores on their leaves called stomata,
Right, stomata.
and the stomata open to allow CO2 to enter. But closing them prevents water loss.
It's a balancing act, right?
Yeah.
Like trying to keep a door open just enough to let in fresh air without letting all the heat escape.
Exactly.
But what happens when water is scarce? Do plants have to sacrifice CO2 uptake to avoid dehydration?
They do. So under drought conditions, they close their stomata to conserve water,
Right.
even if it means less CO2 and reduced photosynthesis.
It's a tough choice, but I guess survival comes first. We were talking about CO2 earlier. How do those C3 and C4 plants differ in their response to CO2 levels?
Well, the C4 plants, with that efficient carbon concentrating mechanism, they are less limited by CO2 availability than the C3 plants.
Okay.
They can maintain high rates of photosynthesis even when CO2 levels are relatively low.
So they're less sensitive to those fluctuations in CO2 levels. That's a pretty big advantage in today's world where atmospheric CO2 is rising because of climate change. What about temperature? How does that factor into all of this?
Temperature is another crucial variable. Just like Goldilocks and her porridge, photosynthesis has this optimal temperature range.
Okay.
Too hot and the enzymes get damaged.
Right.
Too cold and the reactions slow down.
So finding that sweet spot is key for optimal photosynthesis. But we were talking about how those C4 plants are adapted to hot environments. Does that mean they have a higher temperature tolerance?
Yes. The C4 plants, because they can minimize photorespiration, can handle those higher temperatures than C3 plants. They're the heat-loving champions of the plant world.
It's amazing how plants have evolved all these different strategies to deal with different environmental challenges. But we can't forget about water, that essential ingredient for life. We've already talked about how water stress affects stomata and CO2 uptake. Are there other ways that water influences photosynthesis?
Water is involved in so many aspects of plant life, and its impact on photosynthesis, it's often indirect.
Okay.
Besides its role in regulating the stomata, water is also a key component of the electron transport chain.
Right.
Remember those energized electrons
Yes.
zipping along the relay race?
Yeah.
Well, water is needed to replenish those electrons,
Okay.
keep that energy flowing. When water is scarce, that flow gets disrupted and photosynthesis slows down.
So water really is the lifeblood of photosynthesis, essential for both capturing CO2 and keeping those energy conversion processes running smoothly. It's a reminder that all these environmental factors are interconnected, and maintaining that balance is crucial for plant health and productivity.
It is. And as we face a changing climate with more frequent and intense droughts,
Right.
understanding that interplay between water and photosynthesis is becoming even more important.
It's a complex challenge, but luckily, scientists are working hard to unravel these complexities
They are?
and hopefully develop solutions for a more sustainable future.
Absolutely. One exciting area of research is exploring this possibility of engineering C3 plants with C4-like characteristics.
Interesting.
Imagine boosting the efficiency of, say, rice or wheat, giving them those CO2 concentrating superpowers.
Now that would be a game changer.
It would.
It's like giving those workhorse C3 plants a turbocharged engine, allowing them to thrive in hotter, drier conditions.
Exactly. But it's a complex undertaking. Researchers are working to identify those key genes involved in the C4 pathway
Right.
and then figure out how to transfer them into the C3 plants.
It's like delicate genetic surgery, but the potential benefits are enormous. It's a fascinating frontier, combining all this knowledge of plant physiology and genetics and agronomy. But while we wait for these breakthroughs, are there other strategies we can use to optimize the crops we already have?
Of course, traditional breeding, it's played a crucial role in crop improvement for centuries.
Right.
Selecting and crossing plants with desirable traits, breeders have been able to develop varieties that are more tolerant to drought or heat.
It's like a matchmaking service for plants, pairing up individuals with the best genes.
You could say that.
But even with the best genes, those plants still need the right environment to thrive.
That's right. That's where sustainable agricultural practices come in.
Oh, okay.
Optimizing things like irrigation, fertilization, soil management, those can enhance the efficiency of photosynthesis
Right.
and minimize environmental impact. It's all about working with nature, not against it.
It's a reminder that we have a role to play too. By supporting sustainable agriculture and making conscious choices about the food we eat,
Absolutely.
we can contribute to a healthier planet.
We can.
But let's take a break from all the serious stuff for just a minute
Okay.
and indulge in some fun facts about photosynthesis. I'm always amazed by those little quirky details that make science come alive.
Me too.
So what do you have for us?
All right. Well, did you know that photosynthesis is responsible for almost all the oxygen in the Earth's atmosphere?
Wow, so every breath we take is a testament to the power of photosynthesis.
It is a gift from the plant kingdom,
That's amazing.
one we shouldn't take for granted.
Definitely not. Okay, what else? What other surprises do you have up your sleeve?
How about this? The energy stored in fossil fuels, so coal, oil, natural gas, it's ultimately derived from ancient photosynthesis.
It's like a time capsule of solar energy, locked away in those ancient deposits.
It is. And speaking of time capsules, some trees can live for thousands of years, their rings recording centuries of environmental history.
That's right. Those trees are like living libraries, aren't they?
They are.
Those growth rings give us valuable insight into past climates. Scientists can analyze the width and composition of those rings,
They can.
reconstruct historical patterns.
Yeah.
Temperature, rainfall, even CO2 levels. It's like a botanical time machine.
It is.
Allows us to peek into the past,
Yeah.
understand how Earth's climate has changed over time. Amazing how much we can learn from these silent yet powerful witnesses.
It's pretty incredible.
But speaking of change, I'm curious about the future of photosynthesis. As our planet warms, CO2 levels rise, what adaptations might we see?
Well, plants are incredibly adaptable.
They are.
They've been evolving for millions of years to cope with changing environments. I think we're likely to see a combination of things.
Yeah.
Natural selection, some plants might evolve to thrive in these new conditions.
Right.
Others might struggle.
Yeah.
But scientists are working to develop solutions.
Right.
Things like breeding, genetic engineering, those sustainable agricultural practices
Right.
help plants adapt.
So it's like a race against time.
It is.
Plants adapting to a rapidly changing world. And scientists working to develop solutions to ensure their survival, but also their productivity. It's a really exciting time to be in plant science.
For sure.
And for anyone who cares about the future of our planet.
Absolutely.
But let's shift gears again for a moment. I'm curious about how scientists actually measure photosynthesis.
Okay.
Because it's this complex process happening at a microscopic level. How do they quantify it?
Well, there are a few different ways.
Yeah.
And each one gives a different piece of the puzzle.
Yeah.
One common approach is measuring the uptake of CO2
Okay.
or the release of oxygen.
Makes sense.
Remember, those are the key ingredients and products in the photosynthetic equation.
Right, right. So it's like tracking the flow of ingredients and finished products in a factory to gauge its productivity. But how do you actually measure those gases in a living, breathing plant?
Scientists use these specialized instruments. They're called gas analyzers.
Ah, right.
Some gas analyzers measure changes in CO2 concentration in a closed system,
Right.
while others directly measure the oxygen produced using electrodes.
Wow. So technology lets us peek inside these plants, track these tiny gas molecules as they're exchanged. It's amazing. But measuring gases is just one way to quantify photosynthesis. What other techniques do scientists use?
Another powerful one is chlorophyll fluorescence.
Okay, chlorophyll fluorescence.
So remember those excited electrons in the light reactions?
Yes.
Well, some of that energy gets released as fluorescence.
Okay.
It's this faint red glow
Wow.
emitted by chlorophyll.
Huh.
By measuring the intensity and the dynamics of that fluorescence,
Okay.
scientists can learn about the efficiency of the light reactions
Interesting.
and the overall health of the photosynthetic machinery.
So it's like plants are sending out these tiny light signals,
All right.
revealing the secrets of their energy conversion processes. That's incredible. Scientists can use these techniques to study photosynthesis in the lab, but what about out in the field? How do they measure photosynthesis on a larger scale, like in a forest or across croplands?
That's where remote sensing comes in.
Ah, okay.
So you got satellites and airplanes equipped with special sensors that can measure the reflectance of light from vegetation.
Right.
And this provides information about the amount and health of plant life on a global scale.
So it's like taking a giant X-ray of the planet,
It is?
revealing these patterns of photosynthesis across these vast landscapes.
Yeah.
That's some high-tech plant detective work.
It is. And this information is so valuable for things like monitoring ecosystem health, tracking deforestation, even predicting crop yields.
Wow.
It's a powerful tool for understanding the interconnectedness of plant life and the global environment.
It's a reminder that photosynthesis isn't just some laboratory curiosity.
Yeah.
It's this fundamental process that shapes our planet and sustains life as we know it.
It does.
And as we're facing these challenges of a changing climate, understanding and harnessing the power of photosynthesis is more crucial than ever.
Absolutely. And it's a call to action for scientists and policymakers, but also for individuals to appreciate the vital role that plants play and to work together for that sustainable future.
Well said. But before we wrap up this deep dive, let's take a moment to recap those key takeaways for our listener.
All right, quick review. Photosynthesis is the process by which plants, algae, and some bacteria convert light energy into chemical energy in the form of sugars.
Right.
It's a two-stage process. You've got the light-dependent reactions, capturing that light energy, converting it to ATP and an AADPH,
Right.
and then the Calvin cycle, using those energy-rich molecules to fix the CO2 and build the sugars.
It's this complex, beautifully orchestrated process involving pigments, enzymes, electron transport chains, proton gradients, and those remarkable sugar factories we call chloroplasts. And of course, we can't forget the crucial role of water,
Of course not.
not only as a reactant in the equation, but also as the source of those electrons that keep the whole thing going. We've explored all the complexities of photorespiration, the adaptations of C4 plants, and the various factors that influence the rate of photosynthesis. It really is this delicate balancing act between what's going on inside the plant and the conditions it's facing in the environment.
For sure.
It's a reminder that photosynthesis isn't just this biological process. It's this fundamental force that shapes our planet, provides us with food and oxygen, and ultimately holds the key to a more sustainable future.
It does.
And as we continue to learn more about photosynthesis, we're sure to uncover more fascinating insights and innovative solutions to benefit humanity and the planet as a whole. But before we conclude this deep dive, let's leave our listener with a challenge. Something to ponder as they continue their own explorations.
Okay, so listener, here's your assignment. Given what we've learned about these environmental factors that influence photosynthesis, what simple changes could you make in your own life to support plant health and promote a more sustainable future? Could you plant a tree, start a garden, reduce your carbon footprint?
Or simply appreciate the vital role that plants play in our lives. It's a call to action, listener. Let's use our knowledge to make a difference, one step at a time.
And remember, the journey of discovery never ends. Keep exploring, keep learning, and keep asking those questions about the amazing world around us.
It really is amazing to think that something as simple as like planting a tree or starting a garden can actually make a difference for the planet.
It can.
Makes you realize we all have a part to play in this, right?
We do.
But let's not forget the star of our show today, photosynthesis. It's the ultimate life support system, wouldn't you say?
It is, providing us with food, the oxygen we breathe, even the energy we use.
It's true. It's so easy to take plants for granted, but without them, well, life as we know it wouldn't exist.
It wouldn't.
But before we get too philosophical here, I have one more question.
Okay.
We've talked about all the amazing things that plants do, but how do they actually know when to do them? Like, how do they sense the changing seasons, the length of the days, or even just like how much water is available? It's like they have this internal clock and like a sixth sense for the environment.
That's a great question. And it's one that plant scientists are still trying to figure out.
Really?
Yeah. But we do know that plants are incredibly sensitive to their surroundings. And they have this remarkable ability to detect and respond to changes. Light, temperature, water, even gravity.
It's like they've got this whole arsenal of sensory systems.
They do.
Constantly monitoring their environment and adjusting their behavior. But what are these sensory systems? Like, how do they actually work?
Well, let's start with light. Plants have these specialized photoreceptors, molecules that absorb specific wavelengths of light.
Okay.
And when they absorb that light, they trigger all these signaling events
Inside the plant?
inside the plant.
Wow. So these photoreceptors, they allow plants to sense not only if there's light or not, but also the direction,
That's right.
the intensity, and even the color.
Yes. It's like they have these built-in light meters and color sensors.
Wow. That's pretty sophisticated.
It is.
So what about temperature? How do they know when it's hot or cold?
Plants have thermosensors. They're proteins that actually change shape
Oh, wow.
when the temperature fluctuates.
Okay.
And those thermosensors then trigger these signaling pathways that allow the plant to adjust.
So they can adjust their growth?
Their growth, their development, even their metabolism.
So it's like they have their own internal thermometers constantly fine-tuning their physiology to whatever the temperature is.
Exactly.
But what about water? We've talked about how vital water is for photosynthesis, but how do they actually sense when water is scarce and then know to conserve it?
Plants can sense changes in water potential.
Water potential.
Yeah, it's this measure of water availability in the soil, but also in the atmosphere. And they can detect even the slightest changes
Wow.
and respond by adjusting things like root growth, how open the stomata are,
Okay.
all these different processes to keep that water balance.
It's like they have their own internal irrigation system, constantly monitoring water levels and making adjustments.
You got it.
But how do they actually move that water all the way from the soil up to the leaves, especially for tall trees? That seems like a pretty challenging feat.
It is. It's a remarkable feat of engineering.
It is.
Plants have this vascular system that's made up of xylem and phloem, and these transport water and nutrients throughout the plant.
Right.
The xylem, it's made up of these dead, hollow cells, and it acts like a series of tiny pipes carrying water from the roots to the leaves.
So it's like a network of plumbing,
Yeah.
carrying water to all the different parts of the plant. But how does the water actually move upwards against gravity?
It's a combination of factors.
Okay.
Things like transpiration, so that's the evaporation of water from the leaves.
Right.
And then cohesion tension, which is the attraction between the water molecules.
Okay.
So as water evaporates from those leaves, it creates this negative pressure that pulls the water upwards through the xylem, kind of like a chain being pulled from above.
So it's like a giant straw, and the evaporation at the top creates the suction that draws the water up from the roots. That is an incredibly efficient system,
It is.
powered by the sun and the plant's own ingenuity. But we've only talked about water transport. What about those nutrients? How do those get around?
Well, that's where the phloem comes in. It's made up of living cells, and it transports sugars, amino acids, all those organic molecules.
Okay.
Think of it like a network of highways, carrying those products of photosynthesis from the leaves to the roots, the flowers, the fruits.
It's amazing to think about this intricate network, all these transport systems working together, silently and efficiently. It's a reminder that plants are so much more